Hello everybody. I first want to thank the organizers for giving me the opportunity to present our work here at this conference. And I'm also very happy to co-open the proceedings track. The work is about identifying variants in pooled DNA. And here's a short outline of the talk. So first I want to motivate why we're looking at rare variants, and then I want to briefly introduce or give an intuition on the method. Then I present a case study on one of the genes we sequenced, and conclude with a brief outlook also. So we all know genome-wide association studies, and they have been fairly successful. So what they do is basically they have a fixed genotyping chip with, let's say, millions that identifies millions of SNPs, and each of these SNPs usually is considered common. So let's say it has a frequency of 2% in the population, and they have been very successful at finding relevant loci under the common variant hypothesis. So basically, what you do is you take thousands of patients having the same disease, and you take an equally sized or similar sized group of non-patients or healthy individuals, and then you genotype these 1 million SNPs, and then you check whether in one of these two groups you have enrichment of certain genes, so either certain mutations, so either protective ones if they're enriched in the non-patient group, or causal ones which are enriched in the patient group. So the problem is that associated variants often only explain a fraction of the heritability. One prominent example for this is a complex trait. It's human height. And just by checking, do we have a laser pointer? It's not a laser pointer. The mouse. Okay. So by checking this, you can take the mean height of the parents, and you can correlate it with the height of the offspring. And this gives you a fairly good estimate on how heritable human height is. So it's estimated with about 80%. The GWAS identified quite a number of relevant loci. And if you do the same game, so you build a model and you correlate it with the actual height, you get this kind of point cloud, which only explains 5% of the heritability. And so the conclusion also of this is, if it's not a common variance, it must be the rare ones. And that's now how we begin to include the common disease rare variant hypothesis. So maybe just a brief background on this. So if this is a gene and you are looking for something under the common variant hypothesis, you assume that all or lots of patients or sick individuals have the same mutation. With the rare ones, it looks different. So you have it's the same gene. One person actually might have a mutation. Another one might have a stop codon, a newly introduced stop codon. Another one has a splice variant, so the first exon is missing. You have another one with a stop codon or another splice variant or just another mutation. So at the bottom line, you have lots of different variants, and they all cause that this gene is malfunctional, not functional at all, or somehow. And this is what you try to find. So how do you do that? So how do you screen for rare variants? Genotyping chips, as we used them for genome-wide association studies, are not an option here because they focus, as I said, on common variants. So you have minor allele frequencies at the bottom of 2%. This is not enough for rare variants. And most importantly, actually, most of the rare variants are not even on these chips or can't be on these chips because we don't know them. So for these chips, you need first to know the variant before you can find something. So what we can do is we can do sequencing, and that's where high-throughput sequencing kicks or enters the field. So if you're rich, you could do sequencing the full human genome in thousands of individuals, but we know that it's a brute force endeavor to do that. So lots of institutes are involved in this currently in the 1,000 Genomes project. So this is certainly not feasible for a single research site. But what you could do in a medium-sized number of individuals, you could try to enrich exonic regions and try to sequence this and compare that. But if you even want to include more individuals, you have to sequence target regions. And along this line, basically, you have a significant cost reduction in sequencing costs. So focusing on a target region drastically reduces the sequencing cost, but still each sample has to be processed and prepared for sequencing, for high-throughput sequencing. So in a very brief summary, this works like this. You have an individual, you extract DNA, usually from blood samples. You have to fragment the DNA, so you have short bits and pieces, and then comes to the step where you prepare the sequencing library. So this is the step where you ligate adapters to it so that it's fit for sequencing. Once you've done that, you put it on a sequencer. And this has to be done for each of the individuals. So if you have thousands of individuals you want to sequence, you have to carry out the steps 1,000 times. The problem, though, is this step. It's fairly expensive. So actually the major costs are not anymore, if you want to do this, are not anymore the sequencing costs, but it's the cost for the library preparation, ligating all these adapters and making it fit for sequencing. There's a way, there's barcoding, but it only kicks in at a later step. So basically for barcoding, you have in these adapter sequences, you have small identifiers, so you know which sample is from which individual, or which DNA fragment is from which individual after a sequence. But as you can see, you still have to prepare a library for each and every person. So what you can do, what significantly reduces the cost, is working with DNA pools. So here, after extracting the DNA from all the individuals, you take equally molar amounts of DNA from everybody and make just a big pool. And this pool you then process for the sequencing pipeline. So you do the fragmentation, and then basically you only have one library preparation. So now you can sequence, let's say, 1,000 individuals just in one library. And you cut down your sequencing costs by 1,000-fold. And then you do the sequencing. Of course, the reduction at cost and labor comes at a big, big price. So first of all, it's a loss of information. So by doing this, we don't know anymore which subject has which mutation. For lots of studies, this is pretty crucial, but not so much if you just want to screen for rare variants. I mean, if you just want to discover. So it's not a big problem for us. A bigger problem is the increase of noise. So first of all, you cannot do PCR duplicate removal because you have a pool of patients and you don't want to throw away most of the data you generate. And then also, when you're looking for rare variants, which is ideally just one heterozygous allele in your pool, this might be indistinguishable from sequencing errors of the machine you're using. And this is actually the big problem, and it's actually even worse. So the distributors give a sequencing error rate, but this error rate is not uniform, so it varies with what you're sequencing, where you're sequencing, and so on. So now we come to the methods part. So what has been done in the early days, you could, for instance, model sequencing errors as using a Poisson distribution. So you assume errors are fairly random, following a Poisson model. And what you do is you plug in here your expected sequencing errors, so your sequencing error rate in the model. And here's, for a single position that you sequence, you observe your alternative allele count. And then you can compute a p-value, telling you how likely it is that this observed allele frequency was produced by sequencing errors. So as I said before, sequencing errors are not uniform, and here we have a small picture. So this is of one of the target regions we sequenced, and this dashed line shows you the threshold or the cutoff for one heterozygous allele in the pool. And the bars here indicate the minor allele frequency we have, and you see that really lots of bars cross this border, so we would generate lots of false positives. And the colors here identify which actually is the alternative allele. So you see here big stretches of green, so you have a long part where you usually call, I forgot unfortunately the color code, let's say cytosines as an alternative allele. This is also kind of unlikely. But good for us is that these sequencing error rates are quite conserved between different pools or different libraries you sequence, as long as you stay on the same machine, of course. So on the top we have one of our cases pools, and on the bottom we have a control pool. And the peaks align very well, so you have similar error rates at the same position. And also, as you can see, the colors are also fairly the same. So this is all sequencing content dependent on what you have. So this led us to the idea to, instead of looking at each pool individually, we look at combined. So the first step would be just to look at the difference of the two minor allele frequencies. So this is now the plot here. And you have, so now the dashed line basically means that you have one more heterozygous allele in the cases group. So it's the part here, and the dashed line down here means you have one more heterozygous allele in the controls group. And what you see here, and this is the nice effect, your noise, your error rate is completely decreasing here. So this is very, very clean signal compared to what we see before. And so basically these large bars here should be real SNPs. But just looking at the difference and having a crisp cutoff is not really a nice approach. So we thought, well, and how can you do to model the differences of two Poisson distribution? And luckily enough for us, somebody else thought of this. And there's the scalum distribution. And basically you can plug in the same parameters as before. So you put on the expected sequencing error rate of your first library and the expected sequencing error rate of the second library in this position. And then here you put the difference in the observed allele count. And then you can do the same trick and compute a p-value. And if this p-value is small enough, it tells you that it's very likely that one library, so we have a one-sided test only, that one library has one more heterozygous allele in that pool at that position. And in the paper we show that using power calculations we can show by first of all keeping the error rate, the sequencing error rate fixed, that the scalum distribution has more power in finding rare SNPs than the Poisson model. And on the other hand, when keeping the minor allele frequency to be detected fixed, we could show that the scalum distribution is less prone to sequencing errors or less affected by sequencing error rate, the estimated sequencing error rate of the sequencer than the Poisson distribution. And using the scalum distribution, we implemented a small tool in R. And basically currently it focuses only on the most frequent minor alleles so that we don't have to test against all three other alleles. And also it requires, and it's also an important fact, it requires to find a SNP in both in aligned and forward and in backward direction, so that it's also sequencing dependent. And then what we do is we compare one library against all the remaining ones. And so if you have 4 libraries, you take one library and compare it with 3 and see if you find one more higher allele frequency in this first library. And the tool is freely available at SourceForge. So now here we come to the case study. So being in a psychiatric disease research facility, we have a focus on panic disorder. And basically a brief definition of panic disorder is that you experience sudden episodes of acute anxiety and fear occurring without a stimulus. So let's say being at gunpoint is a good stimulus and you're sure it's okay to be afraid, but just walking around here in Vienna is a bad stimulus. So if you feel really afraid, then maybe you have panic disorder. So the gene we focused on is the gene TMEM132D, and it was discovered in a genome-wide association study in our institute. And the gene is fairly large. It covers 834 kilobases of sequence on chromosome 12. And it comprises 8 exons. And so we could not sequence the whole gene because it's pretty long. So we sequenced part of the gene in 600 individuals, 300 cases, and 300 controls. And we distributed these individuals across 4 equally sized pools, so 150 individuals in each pool. And so the regions we wanted to sequence were enriched using custom primers and were focused on the exons of the gene. You can see that down here. So here's the exon structure, and this is the cover which we achieved. And we sequenced on a solid 3+ sequencer. Here's a brief overview on our sample processing. So basically, for each pool, we had 4 pools. We generated roughly 80 million reads. And then we had a quality control and a read trimming step, and we ended up with approximately 2/3 of the reads we had initially. And then everything we had, we aligned against chromosome 12 using Burroughs-Wheeler aligner. And in the end, we had 45 million to 50 million reads for that region for our analysis. And we used the tool vipR for SNP finding. And what we did, we identified 371 SNPs, of which roughly 250 are not listed in SNPDB 132, the latest release. So we had 82 potential SNPs, or potential positions, were validated with an independent technique, the Malditov technique. So this was our gold standard. If we see a SNP using Malditov, we say, okay, it's there. And if we don't see it, we say we had a false positive signal. And the performance of vipR was compared to the 3 other methods. So first of all, the Poisson, which is really related, VARScan, and CRISP. And CRISP is also a method that focuses on multiple pools to find or determine or tell apart SNPs from sequencing errors. So here's how our validation data looks. Basically, for each of the libraries, we have basically one column with SNPs called by our SNP caller, and for these 82 positions, and one column for the MALDI-TOF, and this is basically separate for each of our libraries. And then, for instance, we have, if both callers SNP at a certain position, we have true positives. If there's no SNP in both, we have a true negative. And also we can experience false negatives if only the MALDI-TOF sees something, and false positives if only our method sees something. And this data allows us to compute sensitivity and specificity for each and every library separately. So there's a brief overview on the results. Basically focusing on the left part, we can see that vipR is as sensitive as the other methods. So we achieve a sensitivity of 0.8, same as CRISP, a little bit worse than VARScan, and much better than the Poisson. So what we saw in the power calculation, we could confirm here on real data. And then if we look at specificity, vipR is really top-notch. So it's 0.92, so it's the best of all the methods, and really outperforming VARScan here. So we have better sensitivity at a much more pronounced specificity. So, by varying one of the input parameters of the methods, it's the number of haplotype alleles you have in your pool, and you can run in the same computations over and over again, and you get different sensitivity and specificity values for each of this of the settings, and basically you can end up drawing a ROC curve. And what you see here is that across all the parameters we tried, vipR, that's the blue line, is above the other two techniques. So basically it doesn't really matter which setting you put for the number of haplotypes, vipR produces better results. Here's a brief slide on the runtime. So vipR runs vipR, VARScan, and the Poisson model. They run in an order of minutes, while CRISP is really slow. So this is a log scale. So these are actually days. So for 4 pools, it took on my computer 9 days, which is fairly slow. It's actually slower than the sequencing itself. So to conclude here, sequencing target regions in DNA pools is cost-effective. vipR exhibits good performance in discovering SNPs and even large pool sizes. It outperforms other algorithms in specificity while maintaining comparable, even increased sensitivity. And also this gain in performance is independent of the chosen settings, and it's fast. So here a brief outlook on what is coming up next, or what we're using it for. We're now studying a gene, CLAC16A, which is associated with multiple sclerosis. Basically, it is a disease where where the insulation of your neurons is dissolving and causes weird effects, pretty severe. And it's studied in 200 samples, 100 cases, 100 controls in 4 pools. And we found 300 SNPs, of which 73 are not in SNP-DB. And also we deal with depression. There's recently has been a GWAS study identifying the gene SLC6A15. And here we study 800 individuals, 400 cases, 400 controls in 8 pools. And here we found 505 SNPs. And the majority of them is not known currently. But still, now we're at a point in finding those SNPs, but still it remains a challenge to actually attribute causal functions to everything, to make a sense what's happening there, why these people are sick. So that's in the end of my talk. I just want to thank everybody that is involved in our research institute, so mainly also our statistics genetics group, which I'm part of, then especially Peter Weber, who is running the solid sequencer, and it's really working a lot on this. And then the research group on molecular genetics and affective disorder, Karina Quast and Monika Rex-Hafner, who did basically all the lab work in the validation and so on. And of course our director, Florian Holzberg. And thank you for listening. Thanks.